Okay, so today we are going to study the fluoroquinolones. So before studying the fluoroquinolones, we will see what exactly is the quinolones are. So the quinolone antimicrobials, they comprise a group of synthetic substances possessing in common an N1 alkylated 3 carboxy 4 pyridine ring fused to another aromatic ring which itself carries other substituents. So here is the structure of the first quinolone, that is the nalidixic acid. Nalidixic acid is the first quinolone which is synthesized. So what I have told you, that is the quinolone antimicrobial. They comprise a group of synthetic substances possessing in common an N1 alkylated 3 carboxy 4 pyridine ring. So this is the ring. So this is the ring, that is a N1 alkylated, then 4-oxy 3-carboxy pyridine ring. So this is that ring which is fused to another ring and which is having another 8 substituents. Okay, so this is the structure of the nalidixic acid and which is the first quinolone which is identified. The first quinolone to be marketed was nalidixic acid, which has since been discontinued. So that nalidixic acid, which is identified, now it is get discontinued. The nalidixic acid was classified as a first generation quinolone based on its spectrum of activity and pharmacokinetic properties. The spectrum of activity was limited to a small number of gram-negative organisms. So the nalidixic acid, it is first generation quinolone based on its spectrum of activity and pharmacokinetic properties, and the spectrum of activity was limited to a small number of gram-negative organism. Thus, the quinolone were of little clinical significance until the discovery that the addition of a fluoro group to the sixth position of the basic nucleus greatly increased the biologic activity. So what? That is, these quinolones are having very small activity towards the gram-negative organism. So because of the little activity, these quinolones are discontinued because they are having very little activity against some gram-negative organism. Later on, they modified the quinolones. How they modified the quinolones? That is, they substitute the sixth position of the quinolone with the fluorine group. What they have modified the quinolones? That is, they substitute the sixth position of the quinolone with the fluorine group, and they observed the increase in the biological activity. So the agents that contain the six fluorous substitution are referred as fluoroquinolones and represent an important therapeutic class of the antimicrobials. So this is the difference between the quinolones and the fluoroquinolones. Quinolones are having very little activity towards the gram-negative organism, hence they are discontinued. Then they modified the quinolones with the sixth position with the fluorine group at the sixth position of the quinolone, and they observed that is the fluoroquinolones having increased biological activity. Okay, the norfloxacin was approved for use in year 1986 and represent the first of the second generation quinolones. So there is only one drug in the first generation quinolones, that is the nalidixic acid, okay, which is later on discontinued because having little activity towards the gram-negative organism. Then what they have to do? They did the substitution at the sixth position of the quinolone ring, and they observed the greater biological activity as an antibacterial agent. So the first agent of the fluoroquinolones is the norfloxacin, and it is approved in year 1986, and it represents the first drug of the second generation quinolones. It is considered to be broad spectrum and equivalent in potency to many of the fermentation derived antibiotics. Following the introduction of norfloxacin, intense research carried out, and over 1,000 analogs have now been made. Ciprofloxacin, then gemifloxacin, norfloxacin, ofloxacin, levofloxacin, and moxifloxacin are currently marketed for systemic use in the United States. In addition, ciprofloxacin, levofloxacin, moxifloxacin, and the ofloxacin, along with basifloxacin and gatifloxacin, are available for the ophthalmic use. So these are the various fluoroquinolones derivatives which are available all over the world. And again, these are the structures of the nalidixic acid and the sinoxacin, which are the first generation quinolones, but they are discontinued. Again, why they are discontinued? Because of the smaller activity against the gram-negative organisms. So, what is the mechanism of action of these quinolones or the fluoroquinolones? That is, these are the bactericidal drugs. They kill bacteria by interfering with the DNA replication. How they do this action? That is, they inhibit the bacterial DNA gyrase or the topoisomerase 2 enzyme, and thereby they inhibit the DNA replication and the transcription process. So what is the mechanism of action? That is, they inhibit the DNA gyrase enzyme or the topoisomerase 2 enzyme, and thereby they inhibit the DNA replication and the transcription process. So this is a short mechanism of action of the fluoroquinolones. Now, regarding the structural activity relationship, so this is the structure of all the quinolones. Okay, so this is the basic structure of all the quinolones. So the first SCR point is the essential pharmacophore for activity is the carboxy-4-pyridone nucleus. Yes. So this is the ring. The essential pharmacophore for the activity is the carboxy-4-pyridone nucleus. Okay, so sorry for the interruption. So where we are? Okay, regarding the SR points. So the structural activity relationship. So the essential pharmacophore for the activity is the carboxy 4-pyridone nucleus. So this is what the essential requirement. So that's the first point of the SR of the fluoroquinolones. Then the carboxylic acid and the ketone, they involved in binding to the DNA and the DNA gyrase enzyme system. So these group are essential. That is the carboxylic acid group and the ketone group. So these two groups are essential, as these two groups are involved in binding to the DNA gyrase enzyme system, and thereby they inhibit this enzyme. Then reduction of the 2,3 double bond or the keto group, it inactivates the molecule. If you reduce this double bond or the ketone group, then it inactivates the molecule. Then the substitution at the carbon number 2, it interferes with the enzyme substrate complexation. If we substitute this carbon with any of the functional group, then it interferes with the enzyme substrate complexation. Means what? That is, this molecule does not properly bind to the DNA gyrase enzyme, and thereby it does not inhibit that enzyme. So there is no any substitution at the carbon number 2. Then the fluoro substitution at carbon number 6. So this is the carbon. So if we substitute this carbon with the fluorine group, it improves the antimicrobial activity by increasing the lipophilicity of the molecule, which in turn improves the drug penetration through the bacterial cell wall. So as the topic is the fluoroquinolones, so if there is fluorine group at carbon number 6, then it improves the antimicrobial activity. Then C6 fluoro also increases the DNA gyrase topoisomerase 4 inhibitory action. So the fluorine group is mandatory as we are studying the fluoroquinolones. So the fluorine group at the carbon number 6 is mandatory as it inhibits the DNA gyrase or the topoisomerase 4 enzymes. An additional fluorine group at carbon number 8 further improves the drug absorption and the half-life, but also increases the drug-induced photosensitivity. So if we add one more fluorine group at carbon number 8, it improves the drug absorption and also it improves the half-life. Then substitution of a methoxy group at carbon number 8 reduces the photosensitivity. So the methoxy group at carbon number 8 is found in the moxifloxacin and the gatifloxacin. Then the heterocyclic substitution at carbon number 7, it improves the spectrum of activity, especially against the gram-negative organism. If we substitute the heterocyclic ring at carbon number 7, it improves the spectrum of activity against gram-negative organism. Then the piprazinyl and pyrrolidinil represent the most significant antimicrobial improvement. The piprazinyl group is found in the ciprofloxacin and the pyrrolidinil group is found in the moxifloxacin. They represent the most significant antimicrobial improvement. Unfortunately, the piprazinyl group at carbon number 7 also increases binding to the CNS. Over there, they bind to the GABA receptor, which account for the CNS side effects. Yes, that is the piprazinyl group in ciprofloxacin and the pyrrolidine group in moxifloxacin. They represent the significant antimicrobial improvement. But the piprazinyl group at carbon number 7 of ciprofloxacin, it binds to the GABA receptor, which is found in the CNS and which account for the CNS side effects. Then the alkyl substitution on the piperazine nitrogen in case of the ofloxacin and the levofloxacin is reported to decrease binding to the GABA. So if we substitute the piperazine nitrogen by the alkyl group, then it is reported to decrease binding to the GABA receptor. Okay, so if it is reported that is decreased binding to the GABA receptor, means what? It does not show the CNS side effects. Then the cyclopropyl substitution at N1 appears to broaden activity of the quinolones to include activity against atypical bacteria, including mycoplasma, then chlamydia, and the Legionella species. Okay, so if we substitute the cyclopropyl ring at the N1 position of the fluoroquinolones, then it shows a broad spectrum activity of against most of the bacteria. Then substitution of a 2,4-difluorophenyl at nitrogen 1 improves antimicrobial potency, but agent with this kind of substitution, that is 2,4-difluorophenyl, have been withdrawn from the market due to serious adverse effect. The agents are trovafloxacin and the temafloxacin. Then the introduction of a third ring to the nucleus of the quinolone gives rise to the ofloxacin. So here is the structure of the ofloxacin, and here they introduce the third ring, which is fused to these two rings. Okay, so the introduction of a third ring to the nucleus of the quinolone give rise to the ofloxacin. Additionally, ofloxacin has an asymmetric carbon at the C3 position. Then the S isomer of levofloxacin is twice as active as ofloxacin and 8 to 120 times more potent than the R isomer, resulting from increased binding to the DNA gyrase enzyme. So the S isomer of levofloxacin is more potent than the R isomer, and it binds to enzyme DNA gyrase enzyme more potent than 8 to 120 times. And finally, the last point of the SR is a chemical incompatibility common to all of the quinolones. It involves the ability of this drug to chelate polyvalent metal ions, that is, a calcium, magnesium, and so on, resulting in decreased solubility and reduced drug absorption. Chelation occurs between the metals and the carboxylic acid and the keto groups. So agent containing polyvalent metals should be administered separately from the quinolones. So these agents, whatever the fluoroquinolones, they have the common chelation activity, right? So all these fluoroquinolones, they chelate with all the metals like the calcium, magnesium, zinc, and after chelation, their solubility is get decreased. If the solubility is get decreased, then the absorption rate is also get reduced. And hence these drugs, the fluoroquinolones, they are not administered with the solvents like milk, as the milk contains the metal ions. If you administer the fluoroquinolone with the milk, then the solubility of that drug is get reduced and the absorption rate is also get reduced, right? So these agents containing the polyvalent metals, the agents which contain the polyvalent metals should be administered separately from the quinolones. Means what? That if the doctor have prescribed you quinolones with another agent which contain the metal ions, so that drug which contain the polyvalent metals is administered or get consumed by the time interval. Means quinolone, if you consume at 1 o'clock, then you have to consume the another agent after 10 to 15 minutes. So there is should be a gap interval of 10 to 15 minutes, right? If you administer both the drugs at the same time, then the solubility and the absorption rate of the quinolone is get reduced. Okay. And chelation occurs at the 3 carboxylic acid and the 4 keto groups. So here it is mentioned in the second structure. M means what? That is a metal. So it chelate with the carboxylic acid and the ketone group. And if the chelate are get formed, then the solubility and the absorption rate is get reduced. Then, that is what all about the SR studies. So that is what all about the SR studies. So there are lots of points in the SR. So read it later on. Then the quinolones theoretically fall into one of the four classifications. So here I have mentioned the different classification of the quinolones. So they are classified into the four different classes. That's the first generation, second generation, third generation, and the fourth generation quinolones. So first generation quinolones, I have mentioned you earlier, that is an aldehydic acid and the cyanoxacin. They are the first generation agents, but they are not. They are no more potent. As I have mentioned you, they are no more potent because they don't have the fluorine as a substituent. So first generation agents, that is an aldehydic acid and the cyanoxacin, they are withdrawn from the market. Then the second generation quinolones, they include the norfloxacin and the ciprofloxacin. Now here the term floxacin means what? They are having the fluorine substitution at the sixth carbon. So second, third, and fourth, they contain the fluorine substitution. Means what? The second, third, and fourth generation, they are the fluoroquinolones, and the first generation are only quinolones. Okay. Then the third generation quinolones include the levofloxacin, gatifloxacin, and the gemifloxacin. And the fourth generation quinolones include the moxifloxacin. So this is how the quinolones are classified into the four different classes. Okay, now we are going to discuss with the drugs. There are only for maybe five to yes, there are six drugs. So we will wind up rapidly. So within 5 to 10 minutes, our lectures get finished. So the first drug is the norfloxacin. Again, the IPSA is same for all the drugs. Like yesterday, like sulfonamides. Sulfonamides are also having the same IPC nomenclature. Means if you understand the one IPC nomenclature of one drug, then you can predict the IPC nomenclature of the other drugs also. So the norfloxacin. Now this drug contains the acid. Here it contains the acid. So we have to start from the acid, right? We have to we have to give highest priority to the acid, right? So this is the quinoline ring, and the quinoline ring, the numbering of the quinoline ring start from this nitrogen. Start from this nitrogen. 1, 2, 3, 4, 5, 6, 7, and 8. So this is the numbering of the quinoline ring. So there is a, ethyl group at the first position. There is a ethyl group at the first position. So one ethyl. Okay, one ethyl. After one ethyl, 1, 2, 3, 4, 5, 6. 6 fluoro. One ethyl, 6 fluoro. Then 4 oxo. Then 7 piperazine. 7 piperazine. 1 YL. Why 1 YL? Because uh, the piperazine ring is get attached to the quinoline ring at the first position. At the first position of the piperazine ring. Hence 1 YL. Then the ring is quinoline ring, and the remaining portion there is a 3 carboxylic acid. So this is how we'll nomenclate the norfloxacin. Again, I'll recall you the norfloxacin IUPAC nomenclature. 1 ethyl, then 6 fluoro, 4 oxo, 7 piperazine, 1 YL, quinoline, 3 carboxylic acid. So this is the norfloxacin. Again, in case of ciprofloxacin, the IUPAC is same. What is the addition? The addition is of the cyclopropyl. The addition is what? That is a cyclopropyl. So the IUPAC nomenclature is same as norfloxacin. Here we started the IUPAC nomenclature from the ethyl. Here we start the IUPAC nomenclature from the 1 cyclopropyl. Otherwise, the remaining portion is same. Again, piperazine ring is there, carboxylic acid is there, fluorine group is there. So instead of starting the 1 ethyl, we'll start only with the 1 cyclopropyl. 1 cyclopropyl, and remaining portion is same. So this is what the norfloxacin and the ciprofloxacin. Now the next one is the sparfloxacin. Sparfloxacin. Now here I found the amino group. So we'll start the IUPAC nomenclature from the 5 amino. 1, 2, 3, 4, 5. 5 amino. 1 cyclopropyl. 5 amino. 1 cyclopropyl. Then 7 dash 3,5 dimethyl. Now there is no need to write down the configuration, R and S configuration. There is no need to write down this configuration. So after 7 dash 3,5 dimethyl, then the ring is same. That is a piperazine 1 YL. Then 6,8 difluoro. 6,8 difluoro. In SAR, I have mentioned you something that the fluorine group addition on the 8th position, it is beneficiary to inhibit the enzyme DNA gyrase. Right. So here in the structure of the sparfloxacin, there is a fluorine group, additionally fluorine group on the 8th position. Okay. So 6,8 difluoro, 4 oxo, 3 carboxylic acid. Okay, so this is what the IUPAC nomenclature of the sparfloxacin. What is this IUPAC nomenclature? Again, it is same. Again, the ring is piperazine ring. Ring is piperazine ring. Amino group. What is the addition? If we compare the structure of the norfloxacin, ciprofloxacin, and the sparfloxacin, which additional groups are present over here in the sparfloxacin? There is the amino group, then fluorine group, and 2 methyl groups. This is the additional groups if we compare these 3 structures. Otherwise remaining same. Okay, so now here is the amino group. So we'll start the IUPAC nomenclature from the amino. So 1, 2, 3, 4, 5. 5 amino, 1 cyclopropyl, then 7 piperazine, 7 dash 3,5 dimethyl, piperazine 1 YL, then 6,8 difluoro, 4 oxo, quinoline, 3 carboxylic acid. That is what the sparfloxacin. Now, what is gatifloxacin? Now, in gatifloxacin, instead of the fluorine group on the 8th position, there is a methoxy group. there is a methoxy group. Again, piperazine ring is same, but there is a methyl group on the third position of the piperazine. So, what is the IPS number of the gatifloxacin? So, 1 cyclopropyl, 6 fluoro, 8 methoxy, 7 dash, 3 methyl piperazine 1YL, then 4 oxoquinoline, 4 oxoquinoline, 3 carboxylic acid. So that is what the gatifloxacin. Now this is the structure of the levofloxacin. The IUPAC nomenclature is somehow complicated, so we are not going to discuss the IUPAC nomenclature of the levofloxacin. Just check out the structure of the levofloxacin. Now here, yes, please, please. काढलं. नो नीड, नो नीड, नो नीड, नो नीड. जर का तुम्ही प्लेन बॉन्ड जरी काढले ना तरी चालेल. ते मी जस्ट कॉपी पेस्ट केलं स्ट्रक्चर गुगलवरून, तेव्हा मी काय पाहिलं नाही. तर तुम्ही प्लेन बॉन्ड काढले तरी चालेल. Okay, but don't forget to write down the methyl groups. Okay, means yesterday I have told you that if there is only a bond without any atom or functional group, then you have to understand that there will be a methyl group. There will be a methyl group. Okay, yeah. Now the last structure is the lomefloxacin. Uh, Before lomefloxacin, we are discussing about the levofloxacin. So levofloxacin contain the additional third ring which is fused to these two rings. And the IPC nomenclature of the levofloxacin is somehow complicated, so we are not going to discuss about the IPC nomenclature of the levofloxacin. So just check out the structure. Okay. Now we'll directly come to the last structure of the fluoroquinolones, that is the lomefloxacin. Again, the structure is same. IPC nomenclature is also easy as we have discussed with all the structures. So again, 1 ethyl, 6 8 difluoro, 6 8 difluoro, 7 dash, 3 methyl piperazine 1YL, 4 oxoquinoline, and 3 carboxylic acid. So this is what the levofloxacin. So this is what all about the today's lecture. That is, today we have studied the fluoroquinolones. Hope you have understood the things which I have discussed with you.